microscopic recordings as well. So photoautotrophs can assimilate to their tissues and then grow. The problem with nitrogen is that nitrogen gas really is inert and cannot be taken into the cells as easily. It can be only accumulated actually usually, except in fixing bacteria. It can be only incorporated into the autotrophs by as ammonium or as nitrate. So plants usually pick up nitrate and microbes usually pick up ammonium as well as nitrate depending what's available. Usually, and the important thing about nitrogen in soils, in the water, is that it's a limiting nutrient often. It's not always, but it tends to be a limiting nutrient that limits the growth. And we need nitrogen because it's in the nucleic acids, it's in the proteins, it's in the cell walls. A lot of cell components have nitrogen, so we need it. And it tends to limit the growth, the primary production of microbes and of plants. So that's kind of like the basic difference from the carbon dioxide. A lot of carbon dioxide, a lot of carbon usually go around unless you're in the desert, and but nitrogen, there's a lot of it in the air, but we can't get it, and so there isn't much of it to kind of go around available form. So you need special bacteria that are here that can fix nitrogen to the ammonium. So that's the first step that we're going to talk about, is this nitrogen fixation. And the cycle itself is here, you can assimilate to the amino acids, which is important, and you can mineralize it back to the ammonia. And there is bacteria that can oxidize the ammonium to the nitrate, and there is denitrification in anaerobic environment takes place, and dissonatory nitrate reduction, anamox, back to nitrogen gas or ammonia. So these are the different steps, and it's actually, nitrogen cycle is actually a combination, a dynamic combination of all of these things happening. And in aerobic and anaerobic environments, of course, and not in the same place always. So yeah, so nitrogen fixation. So there is a lot of nitrogen in the atmosphere, and the bacteria. I kind of misled them maybe in the first part of the lectures. I was emphasizing the cyanobacteria as the big group that fixes nitrogen. It's true. They are the most abundant one in the aquatic environments. Actually, they're the most important one in aquatic environments. But there are actually a lot of different bacteria and archaea as well that can fix nitrogen. And so actually 65% of the total fixed nitrogen comes from the terrestrial systems, 20% from marine systems, from cyanobacteria, probably the most dominant one, and 15% is coming from the fertilizers that humans are using to kind of grow our crops. And this is in gigatons. And the one thing about so I said about 15% is fertilizer, and 2/3 is significant amount of food production. So there's war going on right now, and there's fertilizer deficit going on, and that's causing a lot of issues really, because a lot of global food is really relying on the fertilizers. It's a nitrate as well as phosphate fertilizers that we use actually. And with nitrate fertilizers, it's a really expensive thing to kind of make them. Just it requires a lot of energy to fix nitrogen by mechanical means, and often alternatives to the crop management recommended, like grow, for example, like you can go something that can't fix nitrogen in 1 year, and next year you put maybe some plants that can fix nitrogen. For example, here is like soya beans. So beans, usually legumes, tend to have nitrogen fixing bacteria associated with the roots that we're going to talk more closely in the rhizosphere lecture. But so yeah, so so 1 year you could grow something that can fix nitrogen, that will enrich the soil with the nitrogen, and next year you put something that cannot fix nitrogen, and so on. And you can use also wastewater, which has been cleaned but still has high content of nitrogen, and it can be used as to kind of irrigate. Anyway, I prefer my strawberries with whipped cream, not with wastewater. Anyway, so that's one joke sometimes people make. Anyway, so these two guys in 1908, 1910, they kind of invented the nitrogen fixation mechanics. process, Haber and Bosch. There were German scientists. The World War I was like around 1915, 1917. Second World War was 1940. So they were just Germany at that time was not a nice place to be actually. Anyway, that's actually affected their lives quite a bit. So they developed this process. So Haber actually developed the idea, kind of benchtop model, and Carl Bosch was actually the guy who actually took it to the industrial scales. And what else I can tell about that? Yeah, so that model, they both got Nobel Prize because of this process. And so the process itself is combining nitrogen and hydrogen at high pressure and temperature. And today, like currently, like in probably like it's probably not true for all the Earth, but in the human body, it's estimated that like 1/2 of the nitrogen that's in our body actually is from the fertilizers. So if you like organic food, maybe it helps. So it's probably not complete truth, but anyway. But yeah, so do I want to say now the Bosch name you can see on spark plugs sometimes. It's not the same guy, it's uncle. His uncle invented spark plugs. Anyway, so but bacteria have this enzyme complex. Our interest to that process was also because it produces fertilizers and explosives. So I told you about Germany, so the explosives are probably the drive that thing, the interest in this and fixation. Nitrogen, glycerin, and all these explosive compounds. Anyway, so but in prokaryotes there is an enzyme that can do it at room temperature and without the need of pressure in the bacterial cell. And so nitrogenase, that's important. Nitrogenase, that's the enzyme that is able to reduce ammonium, able to reduce nitrogen gas with ammonium. And it's a slow process. It takes quite a bit of time to fix one molecule of nitrogen gas, and it's a huge molecule. It's made of 2 proteins, which has a lot of subunits. It requires molybdenum. Some of them requires molybdenum, iron, and different things, different atoms and metals actually to supplement the enzyme. And so it takes a lot of space in the cell. It's expensive to make, and it's very important that this enzyme is sensitive to oxygen. So this nitrogen fixation by azotropes cannot happen in oxygenic environments. So they need to be somehow protected from the oxygen. So in the textbook that they showed, and I used the paper, says that there is Streptomyces thermotrophicus that can actually do it in the presence of oxygen. But recently came out a paper somewhere in the scientific reports in the Journal of Science, I believe. This one, it says, the title says everything does not fix nitrogen. Okay, so that's kind of like no. There is no known case right now, no known nitrogenase that is not sensitive to oxygen. It's always sensitive to oxygen. So bacteria need to find ways. Bacteria and archaea needs to find ways to kind of get around of that. And the genes coding for nitrogen is highly conservative. So, and it's usually NIF genes, NIFA is usually the 2 dominant ones, but there's a lot of different genes that code for it. You can mine for the NIF genes in the database, and then you know how much you can kind of get. If you're interested in some site and let's say you study and do the microbial sequencing of this one, so you can through metagenomics, not 16S, but through the metagenomics, then you can kind of study how many different species with these genes you could find. You can, if you have time series, you can see if or different based on the treatment plans, different treatment sections of time series. You can see if there is more of this gene. Depending on treatments, you can do qPCR for this gene. So there are different ways to go and use this. These genes are often used to study the nitrogen fixation in different environments or different purposes. And this is the special reaction. I will never ask chemical reactions from you, but it's important. It requires a lot of energy. So it's 16 ATP is required. So it's expensive. And if you have ammonium generated into the in the environment, it inhibits the reaction, of course. So it's kind of like even more of this, and it's used up quickly. Actually, it's in deficit. So yeah. So again, it's not just cyanobacteria that can fix the nitrogen. It's both bacteria and archaea, no eukaryotes. Bacteria and archaea that can fix the nitrogen. And collectively, all these organisms are called diazotropes, and organisms that fix the nitrogen. These are all, and there is at least 100 species of bacteria and archaea known that do that. In case of bacteria, they are in different phyla, but the gene is conservative, so it's kind of has been evolved from one to another, gene transfer or something. In archaea, it's just in one group of archaea that actually has the fixation capability. But in bacteria, they tend to be in different classes and different phyla. And 100 species, almost all environments have them, and they can be ovical or facultative anaerobes as well as aerobes, and hetero and autotrophic, oxygenic or anoxygenic. I say it's sensitive to oxygen, but I will tell you how they can get away in aerobic environments in just a minute. So there's a lot of different species. That's the whole point. You don't need to remember these names, okay? So there are two types of N fixers. So, or actually, there are several types. So, in general, we can say that there is nodulating N fixers in the soils that actually the plant roots sends out some chemicals, flavonoids, to kind of attract bacteria and cause infection, and then they kind of penetrate to the root cells, and the nodules are formed, and this is where nitrogen fixation takes place. So they kind of attract bacteria, they want to be infected by bacteria, and form this symbiosis with the plant roots. And so these are very productive because they fix a lot of nitrogen because they're supported by the plant. So they symbiotic, two guys work better than one guy alone, okay? So one guy alone is doing all the job on his own. If you have two symbiosis, they are much more effective to kind of fix nitrogen. We can fix actually in legumes, rhizobiums, this can fix it's a bacterium. They can fix another one is in Azolla, that's I believe it's a water fern. Should be for the fern, but I'm not sure if it's really biological fern. And it's within these leaves. Anabena is a cyanobacterium, so again, it's in the leaves and they work together, they're much more effective. And the mosses, cyanobacteria, of course, can fix symbiotic again. Free living guys like this cyanobacterium here alone, it's able to fix much less nitrogen per hectare per year or whatever unit you want to measure it. So, yeah. So non-nodulating are these ones that do not produce nodules. So free living cyanobacteria and fixation on aerobic environments. So how so the cyanobacteria usually live they can live in aerobic and anaerobic environments, but they usually live in aerobic environments, and they form these special things, these special cells called heterocysts, and heterocysts have really thick walls, and so they separate physically these cells from rest of the cells which are kind of exposed to oxygen. These ones have anoxic environment, and this heterocysts, they then generate anoxic environment to not let oxygen penetrate the cells, and that's where nitrogen fixation takes place. So these are the cells where they fix nitrogen. And these ones, I think, are called hormogonia. These are, I think, have to do with, or these have to do with surviving not good conditions, like kind of drying, or when environmental conditions are not bad. So heterocysts are these guys. So that's one option. Another option is to use, like I told you about the photosynthesis. The photosynthesis during the day, you fix carbon during the night to respire. So during the night, some bacteria are able to generate anoxic conditions in their cells, and then they fix nitrogen. Then they synthesize this expensive molecule and do this one at night. So this is the red when they do it. And again, everything goes away for daytime when the photosynthesis speeds up again. There are some people who say that they can also do it in if they have mats like that or turfs like that. So within the turfs, in the center of turfs, they can generate anoxic conditions. That has been claimed, and it's also said that mucilage can help decrease the mucilage can decrease the oxygen penetrations to the cells, so that might be one of the ways. And there has been a lot of arguments about the cyanobacteria, the minor ones that are really important in the oceans, like Chlorococcus and Synechococcus, which are the most abundant bacteria in the oceans. And if they can fix nitrogen, and I'm not sure what the consensus is, but I believe it is believed that they are able to somehow, maybe this way or somehow also fix nitrogen. I'm not 100% sure about this, the tiny ones that are really abundant in the oceans. I really don't know about that. Anyway, when the nitrogen is fixed, it's fixed to the ammonium. There is a balance, of course, in the environment between ammonia. It's kind of like it's always like that. It just is like that. They go from one form to another, and of course when ammonia is generated, so ammonium is generated in the nitrogen fixation within the cell, gets assimilated into the cell. It can be used to build up things in cyanobacteria or inside the cell, the isotrophs. These cells can break down and can be used by other organs. So that's how it's kind of gets to the environment. So that's also it gets to iron, of course, by fertilizers can get to the iron, ammonia-containing fertilizers. But there's general environment, there's a balance like that. It's driven by pH, okay? But it's typically there's a balance like that. And yeah, so if ammonia is getting depleted, then there is more ammonia. So there's some sort of balance like that in the environment. And it's ammonia that's actually picked up by cells. And so there are two ways. And really the step to now we're talking about assimilation of ammonia, okay? First assimilation, so how to get into the cell. So there is a balance. So if you take up the ammonia here, there is alpha ketoglutarate. That's an important amino acid. So that's the amino acid that can actually bind to ammonia, and there's and to the glutamate, and that can be used to kind of for other amino acids and transfer this amino group to other places where it's needed. So that first is this balance between these ones. So that's very simple one. There's a balance like that, and but the problem is that so the good thing about this one, it doesn't require energy. So if you have ammonia in cells, you can easily do that. But if you usually do not have ammonia, so you need to use ATP actually to bring the ammonia in. And it's a two-steps process then, and then again you have this glutamate, and from glutamate, the glutamate is synthesized with the ATP, so this amino group is added here, and with another molecule, alpha ketoglutarate, this is split into two, glutamate and glutamate. Two glutamates comes up from these two, but it requires ATPs, two different enzymes. So it's not as easy to do, but these are the two ways to do it. I will never ask you to draw this, but I think it's important to know that this is the amino acids that's getting incorporated in the cells as a first step, kind of assimilate the ammonia. Okay, So ammonification or mineralization is the reverse step. So that's the same reaction. So mineralization when from organics, it can be cleaved, it comes off from the glutamate. That's one of the ways. But outside the cells, usually all these cell walls, proteins, DNA, it's getting consumed by extracellular enzymes again. It's getting split up from the nitrogen-containing compounds are getting split up to the monomers, and from monomers they can be used up by other organisms again. So that's one of the ways. It's just like extracellular enzymes just degrade the organics. That's generally what it says for extracellular enzymes. And here's an example, for example, urea, which is a common one. There's ureases. That can happen also in the cell, but also outside. It's broken up from the urea, like ammonia and CO2. But there's always need for ammonia in the environment, so typically, unless it's wastewater. So this is getting used up quicker. So there is balance between assimilation and mineralization. If nitrogen is limiting in the environment, assimilation is dominant because it limits If there is too much of nitrogen in the environment, ammonification, mineralization takes place. No. Then the cells tend to mineralize, release it from the cells because they don't want to uptake it, doesn't want to assimilate. And the filter from that released to the environment varies. It goes to living biomass, bacteria, and plants. Plants usually do not take really up directly ammonia, they take up nitrate, as I said before. And bound into non-living organic matter or clay, so it can be bound, and it can go through the nitrification process, which is next. So nitrification is a two-step process. So I was talking right now about this step before. I was talking about ammonification, the amino acid, and mineralization back to the ammonia. Okay, so that's what I was telling. Now let's talk about this one, the nitrification process. So this is aerobic, and it's a two-steps process. So first there are microbes that convert ammonium to nitrate, and from nitrate. So they always chemoautotrophs. So they, what else? So they fix carbon side. They generate energy and fix carbon. Not much energy. It's not very efficient way to fix carbon. So there is one. They usually have names like that: nitroso, nitro. These are kind of, you have again your sequencing data and you identify what they are. You can, based on the names, you can kind of figure out what they might be. If you have a lot of nitrosomonas, that means that you have a lot of this process going on maybe in your sample. And there is one species that apparently is able to do everything in one step. So this was recently described. Typically, it's a mixed population. So there is a mixed population of these guys and mixed population of these guys. So everything can go. If you're interested in wastewater treatment, especially pH is important. So wastewater treatment. Okay, I was skipping ahead. We have wastewater treatment lecture, but in wastewater treatment, essentially all this is important up to here, and also this is important up to here. So wastewater really this in wastewater it's critical for nitrogen removal. These steps are important. Everything is important. Depending how many levels of treatment to do. Anyway, so it's important to keep the pH as well as oxygen in your. These are autotrophs, and it requires they requires oxygen for this process to take place. So it's aerobic bacteria. So you need to keep if you want to keep your wastewater treatment going on nicely, you need to have the pH like that. You need to be well oxygenated, but not too oxygenated as well. It just costs too much money. It's waste. If the process goes anaerobic, your wastewater treatment is done from the start from the beginning. Anyway, so that's about. So the required pH is inhibiting, and they are thought to be responsible for acidification of the soils also, because they kind of acidify the environment. It's becoming more acidic. So these are kind of side notes for people who really want to know. There's some chemoheterotroph fungi can also oxidize ammonia but get no energy. The typical eutrophic nitrification is much bigger than this heterotrophic nitrification. But in some soils, these heterotrophs can be important. So there are various bacteria. So everything is not so simple as textbook usually say. And nitrate typically does not accumulate in a natural system because there is no excess of ammonia to begin with. Okay, so this nitrate usually doesn't accumulate; it's getting used up. But it will accumulate if you use fertilizers. If you put a lot of nitrate or ammonia into the system, into the soils, it can accumulate in the crops, not the crops, in the soils, and it's also important process in landfills, septic tanks, and feedlots. And nitrates are negatively charged. Okay, that's kind of important understanding. Some things about both in microbial ecology and water quality is that nitrates are negatively charged, so they are very mobile in soils. Okay, so nitrates move really easily through the soils, and they can easily contaminate ground and surface water. This is very different from bacteria that tends to kind of get attached to the soil particle and like to be attached because they want to leach the organics from this particle and so on, and feast on the particle. So this is big difference between the chemical and microbiological data in water quality actually. The chemical contaminants tend to go through the soils much easier than actually the microbial contaminants, I would say. So the nitrates are mobile, they move through soils easily and can contaminate the groundwater. This is a health concern as well as the eutrophication concern. We have lectures about the eutrophication later. We talk about lakes. Eutrophication is just increase of nutrients like also phosphorus and nitrate, both nitrate and phosphorus, which causes algal blooms, fish kills, and loss of habitat, and the value of your lake goes down. You don't want to swim there anymore. You don't want to sit on a smelly lake, and there's also health concerns. It can cause, in cup, for example, if you drink a lot of water that has a lot of nitrate, the nitrates can move to nitrates, and this can cause this tooth disorder, which is quite a mouthful to pronounce, methemoglobinemia. So we have hemoglobin in our soils, which carries oxygen through the blood into our tissues all over. But these nitrites can actually bind to the hemoglobin and form this methemoglobin, and this one can't carry, and when you can get it affects real babies, can affect cause the blue baby syndrome, and also the nitrites have been speculated. They can form nitrosamines in your gut and can be carcinogens. So that's also has been suggested. So I'm not sure it's 100%, but it's a suggestion that's been made. Okay, any questions about that? It's very depressing, right? Blue babies, eutrophication, and so on. So this is sample of eutrophication. So the result of nutrient-rich water is coming from the Mississippi River, and the area of hypoxia is increasing because of algal blooms that's generated by these nutrients, and when the bloom dies, it requires a lot of oxygen to degrade by aerobic bacteria, and so it's getting it depletes the oxygen, and you get fish kills. Also, you can get so this is Great Lakes. I believe it looks very much like Great Lakes. Fish kills because of no oxygen or because toxins are formed by blue-green algae. I'm not sure if it's blue-green or green. I have no idea. Blue-green. Green algae is. I might be wrong. So we did talk about fixation, about assimilation, mineralization, and we talked about nitrification. Now we talk about nitrate uptake. So to fixing nitrate to the amino acid. So it's a nitrate reduction to the biomass, okay? And now to uptake nitrate. And it must be reduced in cells to ammonia. That's a problem. That requires ATP. That's why bacteria like to use this one, because they don't need to use ATP. If you need to use ATP, it needs to convert it before to ammonia, so that's extra step. Typically, most bacteria prefer ammonium. As I said, as nitrate reduction, ammonia requires energy. Ammonia suppresses asymmetric nitrate reduction. So if there's ammonium, it suppresses this process for bacteria. So it's bacteria prefer to go that way. Okay. Yeah. Sorry, I said twice. Things. Okay. Nitrate reduction. So nitrate reduction, there are two ways. So one is both dissimilar nitrate reduction. It means that it's not assimilated, it's dissimilar. It doesn't get assimilated in tissues. And it's a nitrate reduction to ammonia and nitrogen gas by bacteria, archaea, and fungi, and protozoa. Actually, dissimilar nitrate reduction to ammonium. Okay, so this is the DNRR. This is this one here. This one. This doesn't produce nitrogen gas. This one, the difference is this one is producing actually ammonia, okay? So it produces ammonia, it generates some energy that can be used to do something again. It's by facultative chemical dose. Denitrification or dissimilar nitrate reduction to nitrogen, that actually takes you to the gaseous, to the gas phases, to the nitrogen gas. The problem, it generates a little bit more energy, not much, but produces more energy, so it's actually preferred. The problem with this one is it produces a gas. So it means that final product is nitrous oxide or nitrogen gas, and it goes up to the air and the water, and then eventually the air. So it's going to be, you're losing nitrogen, which is limiting you, so that you're losing this out from the system. So that is a problem with denitrification. So you're kind of losing it from the system. It's on the other hand, it's very good for the wastewater treatment because you want to get rid of the nitrogen. So you remove the nitrogen, and there is limits for the effluent that you can put out in the environment. So you have nitrogen actually released to the, from system, and you're putting out less nutrient-rich water. So this one is the primary, the denitrification, this one, is the primary driver of dissimilar nitrogen reduction in soils, and fungi are very important in it also. Foods, as I said, so it's not good for productivity if you want to run it. And some of these products, like nitrous oxide, they can cause depletion of the ozone layer. And here are several enzymes, so these are the steps actually, and how it takes place. This is kind of, it's a 1, 2, 3, 4 steps, and you have gases, different gases emitted through the process. Requires at least 4 enzymes. And so this is kind of like comparing this and this process, which one is favored. So it depends how much carbon you have. For example, in rumen, which is there is a lot of carbon here, and there is little carbon here. For example, this this is carbon-rich soil. There is not much carbon. So if there is not much carbon, denitrification is dominant. Okay. If there is a lot of carbon in a rumen full with hay, the dissimilar reduction with ammonia is dominant here, okay? So that's kind of like basic which process is dominant in which environment. And they're not inhibited by ammonia, in contrast to this assimilation. And low nitrate levels, so if you have low levels of nitrate, apparently this thing tends to stop here with nitrous oxide. If you have high levels of nitrate, it goes up to the nitrogen gas. That's a textbook that says that. I'm not sure what's the justification of that. So if you have a lot of this, it goes to that. If you have less, it goes to that. This is the nitrate reduction organisms are mostly heterotrophic, using fermented metabolites. Sure, they are anaerobes. While denitrifiers also mostly heterotrophs, but some, okay, that's kind of like exception. Some of them can be actually autotrophs. But they are anaerobes. Okay, climate change. I think we did talk about the methane. So we talked about carbon dioxide, which was important, in the methane. And you remember I told you about that this one is much more potent than the carbon dioxide. Nitrous oxide is even worse, so it's actually much more effective trapping the radiation in infrared, and also importantly, it has a long residence time. So once in the atmosphere, it stays there for a long time. Methane stays only 12 years. This one is over 100 years. So it's degraded much slower than methane. And another problem with that one is it can, in the upper atmosphere, it can be converted to nitric oxide back, which is this one, and which contributes depletion of ozone layer. It's by chemical process, okay? It's not bacteria. It's a chemical process back to the nitric oxide. And this one is involved in depletion of ozone layer, which is bad, of course, because if you have more UV, you get UV inhibits phytoplankton growth, it can cause skin cancer or others. If you go to New Zealand, really, the sun is brutal. The sun in Hawaii is nice and mild. If you go to New Zealand, it's brutal. Even if the weather is cold, the sun can burn you really bad. So yeah, let's see everything about that. I hope so. And anaerobic ammonia oxidation, that's the last process that was discovered only recently, and it's only one group of bacteria that can do it, not archaea, that's a Planktomycetes that can do it, and it's anaerobic ammonium oxidation. So, without going into detail, really, it's important process in actually the oceans. It's also used in the wastewater treatment plant, and Again, like denitrification, it releases nitrogen gas that limits the production. The problem with using this one actually in the wastewater treatment, I believe, is it's really slow growing. So you need to grow something anaerobically in a contained facility and grow slow. So there are freshwater and marine species of Plantomycetes can do it. And I put this one just here for interest because it has the same thing that's here on the left. It's just the same thing here actually. It's nothing different here. But it lists all the genes. I'm not going to ask the genes or enzymes actually. It lists all the enzymes, but there are genes for these enzymes. That these are enzymes, okay, not genes. That involved in these different parts of the nitrogen cycle. And so if you want to start nitrogen fixation, I said you start the NIF gene. If you want the enzyme and the genes involved in producing that enzyme. And so these are kind of like the pointers to the, like what you want to study if you by qPCR or by metagenomics to kind of figure out how the system does. Alternatively, if you study wastewater, you would just study how much you have nitrates or nitrates, nitrates or nitrates in your system, or how much nitrous oxide is produced, kind of tell what's actually is happening. So you don't always need to study the microbes to kind of know what microbes are there and what they're doing. They are nitrogen formed cohorts are metabolically versatile, so some of them can switch depending on what the environment are. So classification of just nitrifiers and denitrifiers actually is believed to be inadequate. It's not as these are distinct steps, but it's not really nitrogen cycle. It's not really just steps. It's a complex network of reactions, and actually new. I think I didn't tell you about one. That there are new ways how the chemicals get transformed, discovered every year, and how the cycles interact. I don't have enough deep knowledge to talk about that, so I say that also how the nitrogen cycle interacts with the carbon cycle, and the microbes actually combine both of these into one source. So it's getting really complicated. It can get really complicated. This network alternate, for example, wastewater accelerate greenhouse gas segregation, human-induced global change. So microbial life harness enormous metabolic diversity is continually uncovered. Several theoretical pathways being explored. Life finds a way, and intervals in Jurassic Part 1. Iron is too whatever necessary to survive. So and challenges to relating nitrogen cycle. I know that there is tendency today to talk that population growth is not the problem. I really don't know. I looked on this graph and it looks like it's a problem. And we want to support so many people. We need food. We can grow crickets, but if we want to grow crops, we need fertilizers to keep it going on the same level that we are currently doing it at least. And the problem is we need a lot of them. The problem is also that we use a lot of fertilizers, but actually not all of it is actually estimated with crops, so we put more than actually is needed, and the result is that nitrogen in our water is includes lower acidification. So there is crop rotation I mentioned, so there's better management strategies that we could use. Crop rotation, optimize timing, and also fertilizer apply, when to apply it, like don't do it when it rains. Breeding or genetically modified organisms improve nitrogen utilization, and develop endosymbiotic end fixers for different plants. So that's ways to go and help actually the crop production. And global nitrogen budget really. So excess nitrogen a lot. So this is a huge amount that is brought into the biosphere each year, and nitrogen cycle is disturbed by human activities. I told you that this is a cycle that has most impacted by human activities compared to everything else. So fertilizers we bring in. So we enhance end fixation by crops. So we grow different crops. We have huge corn fields, not corn fields, huge legume fields maybe. The nitrogen fixing plants that fix the nitrogen. Burning of nitrogen containing fossil fuels, and nitrous oxide is released. So these are all disturbances caused by us and important. And the result is climate change, we have more nitrous oxide in the atmosphere, photochemical smoke, acid precipitation, nitric acid can be formed because of the gases in the air that are produced. The depletion of the ozone layer, I tell you, groundwater contamination by nitrate fertilization, we kind of speed up the global carbon cycle, we add more nitrogen into it, and potential shifts in biological diversity are also potential outcomes that are listed on this paper, okay? I think I put this paper also for you to read if you want to read more about that. And for the US, really, what areas of groundwater are most vulnerable, it's really the areas where we grow our crops and we put the fertilizer, so that's the whole point here, is that the areas where we grow our crops, the middle sections, this is where there's the highest risk for the groundwater contaminations. And there are water quality standards for nitrates and related to human health, so these are the limits that are recommended for drinking water by EPA, which does the American standards, and World Health Organization, that's general recommendations. I think Europe is often using also, but EU has their own systems that I'm not very familiar with, but this is for chemicals, so I'm going to talk about microbes later, but there are limits of nitrogen that can be present. And potential effect, I told you already about these actually all. I told you about the methanoglobinema, blue baby syndrome, carcinogenesis, because the nitrogen, so this is kind of questionable. And it's also said that nitric oxides are kind of very reactive and they can impact various biochemical processes in cell and cause other things that we don't really know. So these are important concepts for nitrogen cycle. Cool. Okay, I got through it. So, any questions about that? No? Everybody's looking on the computer or no? Okay, fine. So I'll get your quizzes back on Monday. So this was just like to kind of motivate you, okay? If you did get point, that's also fine. We do it again sometimes, I'm not telling you when. Today we also